Genetic drift can happen when populations are small, so allele frequencies drift up or down due to chance, like sampling errors when the samples are too small. Now we explore how populations become small through bottlenecks and founder effects in natural populations. The random sampling process that takes place during fertilization occurs in every sexually reproducing population. Similarly, accidents that remove individuals at random occur in every population in every generation. It is important to realize, though, that since drift is caused by sampling error, it can occur by any process or event that involves sampling. Not just the sampling of gametes that occurs during fertilization or the loss of unlucky individuals due to accidents. Let's consider two special cases of genetic drift called founder effects and bottlenecks. When a group of individuals immigrates to a new geographic area and establishes a new population, a founder event is said to occur. If the new population is small enough, then allele frequencies in the new population are almost guaranteed to be different from those in the source population due to sampling error. A change in allele frequencies that occurs when a new population is established is called a founder effect. In 1995, fishermen on the Caribbean island Anguilla in the Caribbean witnessed a founder event. A few weeks after two major hurricanes swept through the region, a large raft composed of downed logs tangled with other debris floated onto a beach in Anguilla. The fishermen noticed green iguanas on the raft and several that made it to shore. Since green iguanas had not previously been found on Anguilla, the fishermen notified biologists. The researchers documented that at least 15 individuals had arrived. 2 years later, they were able to confirm that at least some of the individuals were breeding. A new population had formed during this founder event. It's unlikely that allele frequencies in the new Anguilla population of green iguanas matched their source population, which was thought to be on the islands of Guadeloupe. Colonization events like these have been the major source of populations that occupy islands all over the world, as well as island-like habitats such as mountain meadows, caves, and ponds. When a founder event occurs, the founder event is likely to accompany it, changing allele frequencies through genetic drift. If a large population experiences a sudden reduction in size, a population bottleneck is said to occur. The metaphor comes from a few individuals of a population fitting through the narrow neck of a bottle by chance. Disease outbreaks and natural disasters or other events can cause population bottlenecks. Genetic bottlenecks follow population bottlenecks, just as founder effects follow founder events. A genetic bottleneck is a sudden reduction in the number of alleles in a population. Genetic drift occurs more prevalently during genetic bottlenecks and causes a change in allele frequencies. For example, from a natural population, consider the humans in the Pinjalop Atoll in the South Pacific. Only about 20 people out of a population of several thousand survived a typhoon that occurred 250 years ago. Survivors included at least one individual with a loss of function allele at gene CNGB3 involved in color vision. The CNGB3 allele is recessive, and when it is homozygous, it causes the serious vision deficit, achromatopsia. People with this condition have poor vision and are significantly colorblind, as shown in the left picture. Extremely rare in most populations, the frequency of the CNGB3 allele is estimated to be under 1%. In the population that survived the Pinjalop Atoll disaster, however, the loss of function allele was at a frequency of about 1/40th, or 2.5%. If the allele was at the typical frequency of 1% or less before the population bottleneck, then a large frequency change occurred during the bottleneck due to drift. In today's population on Pinjalop Atoll, over 1 in 20 people are afflicted with achromatopsia, and the allele that causes the affliction is at a frequency of well over 20%. The loss of function allele is extremely unlikely to be favored by directional selection or heterozygote advantage, so researchers hypothesize the frequency has continued to increase over the past 230 years due to drift.